Okay, my name is Lei Du. I come from Northwestern Polytechnical University. Today, I want to talk about our works titled Identifying Diagnosis-Specific Genotype-Phenotype Associations with Joint Multitask Sparse Canonical Correlation Analysis and Classification. I will first introduce the background and motivation. After that, I will present the details of the diagnosis-specific multitask SCCA and logistic regression model. Then I will show you the experimental results on both simulation data and real data. And finally, I will give a conclusion. In this study, we focus on the brain disease, so I will use Alzheimer's disease as an example in this talk. In traditional bioinformatics studies, if we want to find out the AD risk genes, we can do the following things. We first collect the gene data of a group of people with healthy control, mild cognitive impairment, and Alzheimer's disease. Then we can build a classifier or a prediction model with feature selection to detect which gene contributes to the Alzheimer's disease. But researchers have found that using a discrete description of the brain is insufficient because the brain status is continuous, not discrete. So they propose to use some continuous biomarkers to describe the brain accurately. Then we can collect the neuroimaging data by the MRI scanner or the PET scanner. This neuroimaging data can describe the brain continuously, which can improve the detection power. So the goal of imaging genetics is to investigate the role of genetic variation in disordered brain function using neuroimaging biomarkers as phenotypes. This is a popular framework for imaging genetic association studies. We first collect the gene data of a group of subjects. Then we collect the neuroimaging data of the same groups. Then we can build a bimultivariate association model, such as SCCA, to find out the bimulti association between SNPs and neuroimaging markers. Because we usually want to find out a small subset of SNPs and brain ROIs, so the sparse learning techniques is also used. For example, the FD block guided group lasso and the brain circuitry guided graphical lasso are usually used to guide the feature selection for both SNPs and brain ROIs. But this model has some problems. First, different association directionality for different diagnosis groups. For example, HCs and ADs could carry different imaging and genetic markers. Different diagnosis groups may have different risk losses. Different AD subsets, for example, early onset and late onset AD, may have different imaging and genetic markers. Third, SCCA is unsupervised. This means the diagnosis information is usually overlooked. The three problems cannot be addressed by the model we just presented. So, we want to overcome these three problems by a novel diagnosis-specific multitask SCCA and logistic regression. This is the framework of our method. We first split the diagnosis group into different subgroups. In this study, we split a group of subjects into three groups such as HC, MCI, and AD. Then we conduct class binarization and class balancing to generate three different datasets, HC versus non-HC, MCI versus non-MCI, and AD versus non-AD. This three dataset is different but holds some similar pattern. So we use the multitask SCCALR model with feature selection, and finally we yield three feature sets. They are HC-specific, MCI-specific, and AD-specific SNP-QT associations. In particular, we use X to denote the gene data, Y to denote the neuroimaging data, and Z to denote the diagnosis information. We first repeat XYZ for 3 times, and then we use class standardization and class balancing to generate 3 different subsets. They are X1, Y1, Z1 for HC versus non-HC, X2, Y2, Z2 for MCI versus non-MCI, X3, Y3, Z3 for AD versus non-AD. Then we use one versus all modeling strategy to build the multitask model. Then this model can help us to identify the diagnosis-specific feature sets. This is the objective of our model. Our model has 3 advantages. First, the LR logistic regression objective encourages the diagnosis-specific feature selection. The LSCCA objective encourages the imaging genetic association identification, and 2 penalties omega U and omega V encourage the diverse feature selection to find out the diagnosis-specific feature subsets. Here I will show you the details of omega U and omega V. Both penalties have a diverse feature selection capability. They can find out both shared patterns and specific patterns. For omega U, we first use FGL norm to help identify the LD block within the SNP data. Then we also use the L21 norm to find out those SNPs shared by all diagnosis groups. The L11 norm on the vector U to help find out those SNPs that only held by a specific diagnosis group. The first penalty for the omega V is the graph-guided lasso. It penalizes every two nodes on the graph to help find out those correlated network nodes. This can help us to find out those related brain areas across the brain. We also use the L21 norm on V to find out those brain areas shared by all diagnosis groups, and L11 norm for V to find out those brain areas that only held by a specific diagnosis group. Since our objective is biconvex, we can use the alternatively iteration algorithm to solve the model. We first fix U to solve V, and then we fix V to solve U. These two steps repeat until convergence. Now I will show you the experiment. We use both simulation data and real data. We simulated 4 simulation data. The first 2 datasets have the same ground truth but different noise. The third data simulates a task-specific situation, and the fourth data simulates a task-consistent situation. In the real data, we have 755 participants, where we have 182 HCs, 292 MCIs, and 281 ADs. We use 1,692 SNPs near the ApoE gene, which is a well-known AD risk gene. We use the AV45 PET scans, which measures the amyloid depositions across the brain. According to the AL Atlas, we divided the whole brain into 116 brain ROIs. This figure shows the feature selection results on the simulation data. We can clearly see that our model can identify class-specific features, but DSCC and GSCC cannot. They can only find out diagnosis-consistent feature subsets. This two figure shows the canonical correlation coefficients and classification accuracy. From the testing CCCs and classification accuracy, we can see that our method have better or comparable CCCs and classification accuracy compared to DSCC and GSCCA. This figure shows the feature selection results on the real data for the neuroimaging data. We can see that both the SCCA and GSCCA can only find out consistent feature subsets. But our method, we can find that it can identify diagnosis-specific imaging QOTs across different diagnosis groups, and within each group there is a strong grouping effort. This is very meaningful. And an interesting result is that for HCs and ADs, our method identified opposite directionality for the feature subsets. This is quite meaningful because our method can tell us that ADs and HCs have different imaging genetic association directionality. This is the identification result of the SNPs. We can also find that DSCC and GSCCA can only find diagnosis group consistent SNPs, and our method can find out different SNPs for different diagnosis groups. This is very meaningful. This is the testing CCCs and testing classification accuracy also demonstrate that our method have better or comparable results compared to DSCC and GSCCA. This table shows the top 10 selected diagnosis group specific imaging markers. We found that all the top 10 imaging markers are AD risk. This demonstrates the effectiveness of our method. This table shows the top 10 selected SNPs for our method. We can also find out that all these top 10 SNPs are AD risk. It's interesting that our method can find out the opposite sign for the feature subset for ADs and HCs. This means our method has strong feature selection capability than both DSCC and GSCCA. Now let me make a conclusion. We first propose a novel diagnosis-specific multitask SCCA and logistic regression method. We also provide an efficient optimization algorithm, and our method can identify opposite SNP QT associations for HCs and ADs. Our method can also select meaningful diagnosis-specific feature subsets. This is quite meaningful than the traditional SCCA method. Now come to the end of my talk. Thanks for your attention.